And it's a little bit of a play on words because the kinds of molecules that we study in my lab are sugar molecules. But they're not the type of sugar that you might think of on a daily basis, like sugar in the food that you eat. It turns out that in biology, sugars have a really important role as a coating on the surface of cells. And so when I describe how these sugars relate to the cell, I often like to start with the analogy to the peanut M&M, because not everybody here is familiar with cells and the structures of cells, but everybody has probably encountered the peanut M&M. And as you know, those M&Ms are covered with a sugar shell. And when I was in college, so this brings me back to the 1980s, I remember learning this in a biology class. And somebody raised their hand and asked the professor, well, why? Why are cells covered in sugars? What's the purpose of that? And back in the 1980s, people didn't really have a very good idea as to what the actual function of the sugar coating was. In fact, people just assumed it was no more interesting than the sugar coating on the M&M. And people of a certain age, which maybe some of these parents and family members are, might remember the M&M commercials from the 1980s, which was that M&Ms melt in your mouth, but not in your hand because of that protective coating. And so that's what people thought about cells, that they just have this protective coating. Well, fast forward 60 years, and we now know that the sugar coating on cells is, first of all, far more complex in terms of the structures of these sugar molecules, and that they serve a lot of really interesting functions. So nowadays, if somebody was trying to use a metaphor to describe that sugar coating, instead of the peanut M&M, they might think of those sugars like all the foliage on the surface of the planet Earth. I think that's probably a better representation. And just like the trees and the shrubs and the grass on the plains that you would see if you were flying over the planet in a helicopter, you would see that these are complex branched structures that are moving and swaying. And there are patches on the planet Earth where you have flat plains of grass and other patches where you have a forest of trees. And the sugars on the surface of your cell are like that too. They come in lots of different shapes and sizes. So a textbook might depict the surface of the cell more like this, where all of our cells have a structure. We call it the plasma membrane. That's a little bit like the chocolate layer in the peanut M&M. It's kind of a layer of fat that encases our cells. But then attached to the surface of that plasma membrane are these long, complicated, and branched polymers. And they're basically, we call them glycans nowadays. But glycans are polymers built from simple sugar building blocks. So in this particular cartoon, these beads on a string, this would be a glycan structure where each of the individual beads was a simple sugar building block, and they're all linked together in these patterns. It turns out the patterns are not arbitrary. Different types of cells have distinct patterns of these glycan structures. And we can actually read out the structures to determine whether a cell is, for example, a blood cell or a muscle cell. There's a signature there. And what got me interested in this field, which really started when I was doing my PhD training back in the late 1980s, was the observation that not only are there sugars that are distinctive on different cell types, but those patterns change when cells go from being in a healthy state to being in a diseased state, especially in the setting of cancer. So I remember reading some articles that were published actually probably in the 1970s and early 1980s that were describing some of the changes in these glycan structures. And one of the changes that seemed to be quite common in the setting of cancer was a change in the abundance of glycans that have a particular sugar building block called sialic acid. It's one of the building blocks that would be in that beads on a string structure. The name sialic acid makes it sound like this is not a sweet sugar, but maybe something more like an acid, like a vinegar. But that's just a kind of a chemical terminology. A sialic acid is a sugar molecule. And when people who study sugar biology try to describe these structures in cartoon form, we often use these kind of lucky charm shapes. So some of those building blocks are squares, and some are circles, and some are diamonds. And sialic acid, we always show with this little symbol of the pink diamond. But if anybody who's tuning in to the Zoom call has an interest in chemistry and what molecules might look like in terms of their atomic representation, it would be this chemical structure here. And this, for example, would be what I would teach your students if they were taking my class in organic chemistry. I would teach them how to draw the structure of a sugar like sialic acid. So the observation that is now more than half a century old is that if you look at healthy cells from your body, you'll see a particular pattern of these complex sugar molecules that have a sialic acid, and it's usually right at the tip of a branched structure, like the leaves on a tree, for example. And that's a healthy pattern, and I like to think of that as like a well-manicured garden of sialic acids. But then if you compare the pattern of the healthy cell to cancer cells that are in a tumor in that tissue, whether it's a lung cancer, a breast cancer, a colon cancer, you can identify the cancer cells from a biopsy, a surgical biopsy, and there's ways of looking at the density of sialic acid. And what people found is that that well-manicured garden in the healthy cells had turned into like a tropical jungle of sialic acids on the cancer cells. And this was a common observation that seemed to get worse and worse as cancers became more aggressive or as tumors grew larger. But what nobody really understood until maybe 10 or 15 years ago is why? Why does this happen? And if we can understand why this happens, maybe there's some knowledge there that might be useful, either for the detection of cancers, diagnosis of cancer, or maybe even for treatment of cancer. So the students in my lab started working on this problem. And to make a long story short, what we discovered is that it has to do with the important role that our immune system plays in protecting us from cancer. So your immune system involves what we call the white blood cells coursing through your blood vessels. And so if you ever get cut and you see your own blood or give a blood draw, it looks red. That's because of the red blood cells. But there's also white blood cells in there that are harder to see. And those are your immune cells. And what your immune cells do, like for a living, their job is to circulate throughout your body and sample all of your other cells one at a time to determine whether those cells are good, healthy cells or bad, damaged cells. And a cancer cell is a damaged cell. So when things are working well in your body, your cells that get damaged, that might be at risk of becoming a cancer cell, those damaged cells will basically put molecules on the surface of the cell to communicate to your immune system that there's a problem and that the immune cells should come and get rid of that damaged cell. They should literally kill it and get rid of it before it can do you some harm. And the way this works is those immune cells, which are these green, hairy, ball-like structures, this, by the way, is an actual microscope picture of immune cells attached to a cancer cell. And what those immune cells are doing is they have molecules on their surface that are interacting kind of like a handshake with molecules on the surface of the cancer cell. And that immune cell is literally tasting the surface of the cancer cell through all of these handshakes and trying to collect information to make a decision about whether this is a good guy or a bad guy. If it's a good guy, then those immune cells will say, okay, nothing wrong here, move along to the next cell and taste that cell and make a determination. But if the immune cell senses that there's a problem with that cell that it's tasting, it will launch an attack. And so if you kind of zoom in on what's happening at the point of contact between the immune cell, so depicted with this sort of half circle, that's landing on the surface of that cancer cell, it turns out it's complicated. There are actually two different types of molecules on the surface of the immune cell. We call them receptors, but they're basically hands that are reaching out to kind of shake hands with something on the cancer cell. One set of those receptors are what we call activating receptors. They're shaking hands with something on the surface of the cancer cell that has been put there as a signal that I'm damaged, please kill me. And so if there's enough handshaking between the green molecule and the blue molecule, then a signal gets sent inside the immune cell that causes it to react and literally tells it to drop a bomb and kill that cancer cell. Now, at the same time, and it seems a little paradoxical, there are other receptors on immune cells that literally do the opposite. And those are what we call inhibitory receptors. Those are different kinds of hands that are looking to shake hands with a different kind of signal, because our normal healthy cells have things on the surface that tell immune cells, hey, I'm good, I'm healthy, I'm fine, move along. And these inhibitory receptors, which are often referred to as immune checkpoints, those were only discovered like 20 years ago. This is a pretty new discovery in the 2000s in the field of immunology. What those inhibitory receptors do is they collect information on the healthy signals, and if there's enough of that information, they will literally overpower these activating signals and block the reactivity of the immune cell, so it won't launch a bomb. And you might ask, why would we do this? Why would we have these two different ways that deliver opposite messages to the immune cell? And the reason is that the immune cells have a pretty tough job. They have to figure out which of your cells are damaged and should be killed from which of your cells are healthy and should be not. And sometimes there's a pretty fine line between the two. So these inhibitory receptors are meant to protect healthy cells so they don't get accidentally killed. Because if your immune system starts killing healthy cells, you'll get an autoimmune disease. That's literally what autoimmunity is. It's the inadvertent killing of healthy tissue by your immune system. And so this checks and balances system is meant to kind of, give the immune system a good shot at making a good decision about who to kill and avoid making a bad decision. And the big discovery in cancer biology in my lifetime, I would say, is the fact that when cancers succeed, it's usually because they have figured out a way to trick immune cells into thinking they're totally healthy. And the way they do that is by increasing the abundance of these orange healthy signals so that they will dominate over these blue damage signals. Now, when all of that came to light, it gave people an idea for how you might be able to treat a patient with a drug that blocks the inhibitory signals so that the immune system can do a better job at recognizing a cancer cell and killing it. And the drugs that block inhibitory receptors so that you don't get this inhibition of the immune cell are what we call the first generation immune therapies or cancer immune therapies. And these immune therapies really revolutionized the practice of oncology. Just to give you a sense of, why people got so excited about the discovery of checkpoint inhibitory receptors and drugs that block them, I'll show you what our field considers like an iconic image from a clinical trial testing two of these first-generation immune therapies. This is what we call a Kaplan-Meier curve, but it's a typical way of showing the data from a clinical trial in which cancer patients were treated with experimental medicines, and their survival was charted as a function of time. This is actual data from a phase 3 clinical trial for patients that had advanced metastatic melanoma, which is skin cancer. And prior to the advent of immune therapies, this type of cancer was considered 100% fatal. There was no way that a patient could survive metastatic melanoma. So that's why it was so shocking when patients were treated in this particular trial with therapeutics that block, and these are just technical terms, but one of these checkpoint inhibitory receptors is called CTLA4. The other one is called PD-1. I apologize. Scientists have a lot of acronyms and jargon. I try to minimize that. But these are two of the most important checkpoint receptors that were discovered 2 decades ago. And they were blocked by drugs that are monoclonal antibodies. These are protein therapeutics. They look like this in terms of their shape. They look like kind of the letter Y. They're proteins that get administered to a patient by IV infusion. And in this trial, patients were treated with one of those drugs, which is the green curve that targets the CTLA4 checkpoint receptor. Some patients were treated with a different drug, which blocks a different T-cell checkpoint receptor called PD-1. And then a third group of patients were treated with a combination of the two drugs. And you can see that some of these patients did not survive, but at the 3-year mark, so 36 months into this trial, almost half the patients that were treated with either the single drugs or more than half with the combination therapy were still alive. That was unprecedented. And those patients, they kept living and living and living. And some of those patients are still alive a decade later. They were essentially cured of what was otherwise a 100% fatal disease. So that was really stunning, and it really blew open the field of cancer immune therapy and ultimately led to the development and approval of a whole suite of immune therapy medicines that target these two checkpoint receptors, either the CTLA4 receptor or the PD-1 receptor. And you're looking at a collection of those medicines now. Basically, every major pharmaceutical company that has a portfolio in oncology will have an immune therapy that targets these inhibitory receptors. And you're seeing some of the big blockbusters here on this slide. But as exciting as that was, it didn't solve the entire problem. And which isn't to say that these weren't absolutely groundbreaking medicines. You can see that at least for half the patients that were treated in this melanoma trial, they benefited, they survived, some of them were cured. But the other half did not enjoy such a benefit. Nonetheless, to cure anybody with metastatic melanoma was a major breakthrough. So I think it's totally reasonable that back around the time these drugs were approved, which is around 2012, 2013, so over 10 years ago now, all the major science magazines and journals were hailing immune therapy as a revolution in cancer. And maybe we were going to cure cancer after all, which has been one of the moonshots, for most of my lifetime. In fact, some famous people were cured. Former President Jimmy Carter had metastatic melanoma in his brain. He was diagnosed, around 2011, and he got into a clinical trial for one of these immune therapies, and he was cured, lived another more than a decade, and only passed away just last year, over the age of 100. And the scientists who discovered the checkpoint receptors, these two immunologists, were recognized with the Nobel Prize in Physiology or Medicine back in 2018. So again, these are revolutionary breakthroughs, but as great as it is for half the melanoma patients, the other half did not benefit from these drugs. And furthermore, in other clinical trials for different types of cancer, these drugs have been helpful for even fewer patients. So here are some examples of the percentage of patients that benefit in trials for a certain type of lung cancer or bladder cancer, gastric cancer. And you can see the number of responding patients is even lower. So the big question in the field over the last decade has been, what are we missing? Why is it that some patients, usually less than half, can be cured with this particular class of immune therapies, but still the majority of patients don't seem to respond? Their immune systems remain paralyzed against the cancers. So the thought here is that there must be other receptors and other, handshakes going on at that point of contact between the immune cell and the cancer cell that don't involve these particular receptors, the CTLA4 and the PD-1 receptor, which are two proteins, by the way. So this is where I'll come back to this long-standing observation that one of the big differences between tumor cells and healthy cells is this tropical jungle of sialic acids, this difference in the sugar coating. And, most of the people in the cancer immunology field don't really think about sugar biology. Sugar biology is a fairly small niche field in biology. It's much more common for people to focus on the biology of other kinds of molecules, like proteins or nucleic acids. But in my lab, we had been thinking about the sugars and studying them in their various biological processes. And it occurred to us that maybe the missing link here has to do not with proteins, but with sugars. And the reason we hypothesize that is that around the year 2000, you might remember, the human genome got sequenced for the first time. And since then, millions of human genomes have been sequenced. And as soon as our genomes got sequenced, we got insight to all the different molecules that our cells are able to produce. And one set of molecules that was discovered through the human genome sequence was a family of receptors on immune cells that bind that sugar, sialic acid. There's an acronym, like everything in science, for these receptors. They're proteins that are anchored to the plasma membrane, so they're on the surface of the immune cell. And we call them the sialic acid binding, Ig, which stands for immunoglobulin, like lectins. And that's just a big mouthful. So the acronym we use is Siglec. These are the Siglec receptor family. In this cartoon, the yellow line reflects the surface plasma membrane of the immune cell. And all the blue stuff is what's outside the cell, facing outward. And all this stuff down here, these red bricks and purple bricks, this is what's inside the immune cell, okay? And first of all, I'll point out that there's 14 members of the Siglec family in humans. There's some funny numbering here because the numbers came from mouse Siglecs and they're a little different in humans. They're not just on one type of immune cell. By the way, I didn't mention this earlier, but there's lots of different types of immune cells. Our white blood cells are kind of like the US military industrial complex where, we have the Army, the Navy, the Air Force, the Marines. And likewise, in the immune system, we have the T cells, the B cells, the macrophages, the neutrophils. There's all different types. And every single type of immune cell has one or sometimes more than one Siglec receptor on it. Okay, so they're all over the entire military industrial complex of our immune system. These red bricks and purple bricks mean something. These are little motifs within the proteins that are actually known, and quite famously known, for being involved in that inhibition signal. And so this is what kind of triggered our imagination here, because when we looked at these motifs, these purple bricks and red bricks on this inside the cell part of the Siglec receptors, we realized that that's very similar to what you find inside the cell in that checkpoint receptor PD-1 that people had been targeting with blocking monoclonal antibodies as drugs, and that had worked, for less than half of the cancer patients. And so the parlance we would use is that what's inside the cell on the Siglec receptors is very similar to what's inside the cell on PD-1. So they have a common signaling machinery that inhibits the activity of the immune cell. And so this is what got us to analogize that maybe the Siglec receptors are kind of doing the same thing to the immune cell that this T cell checkpoint receptor is doing. And if that's true, it would make sense that a successful cancer cell that was trying to hide from the immune system and protect itself might want a tropical jungle of sialic acids that can engage these receptors in a handshake. Okay, so this is the cartoon that describes our hypothesis. We thought this well-known biology, this Nobel Prize winning biology, wherein the T cell has a checkpoint receptor called PD-1 that binds to a ligand on the cancer cell, that's the handshake, the checkpoint ligand, that delivers inhibitory signals to the T cell. And these blocks inside the cell are involved in that inhibition. Maybe likewise, the reason cancer cells would have all of these sialic acids is because that would allow them to latch onto the Siglec receptors and basically do the same thing, except not just to a T cell, but to all the different members of the immune cell military industrial complex. So we tested this hypothesis experimentally in a number of ways, and I won't go through all the technical details, but the bottom line is this. What we found is that when things are working well for you, your immune cells will shake hands with a cancer cell. That gives a bad taste and tells the immune cell, I'm going to kill. However, if the cancer cell, just through random luck and natural selection, happens to figure out how to coat itself with a tropical jungle of sialic acids, that cancer cell now can shake hands with one of the Siglec receptors, or maybe multiple Siglec receptors. And that tells the immune cell, hey, that cell tastes good. I don't think it's a cancer cell. No bombs, lay off, move along. So natural selection would allow the cancer cell to thrive as long as it had a coating of sialic acid. Well, this is where we thought, all right, this might be why so many patients are not responding to the T cell checkpoint blockade. Maybe those are the patients that have so much sialic acid, they don't need the checkpoint receptors. They have their own way of hiding from the immune system through the Siglec receptors. And if that's true, maybe we want to somehow block the handshake between the Siglec receptors on the immune cell and the sialic acids on the cancer cell. So we thought about how do we block that handshake? Now, taking a page out of the playbook of those successful checkpoint receptor drugs, the PD-1 blocking drug, the CTLA-4 blocking drug, maybe we should also just make a monoclonal antibody protein that can attach itself to one of those hands and block it so that it can't shake hands with those sugars on the surface of the cancer cell. Okay, that would be a very similar strategy that worked, for all of those other first-generation immune therapy drugs. But the problem is, there's not just one Siglec receptor. There's a whole family of them. And what we discovered is that it's impossible to make one drug, one monoclonal antibody drug, that can shake hands or block the hand of all the members of the Siglec receptor. They're not similar enough to each other to have a common drug. And we don't know if there's one that we should block that's more important than some of the others. That knowledge isn't available yet, although we're working on that. But in the meantime, since we don't know which Siglec to block, or if we have to block multiple Siglecs, we thought, let's take a different strategy. One thing we know is that all of those 14 Siglec receptors, they're looking to shake hands with a sugar that has sialic acid on it. That sialic acid is essential for the handshake. And so we figured if we could get rid of sialic acid from the surface of the cancer cell, then that would basically interrupt all of the handshakes with all of the Siglecs. So that became the focus of our drug discovery activities, which have been ongoing now for the last 9 years or so. And the idea we had is to make a new generation of immune therapies that are basically like lawnmowers. The idea was, let's make a drug that parks on a cancer cell, not on a healthy cell, but just on a cancer cell, and that will drive around on the surface of the cancer cell and literally cut the sialic acids off and throw them away. And once you strip the sialic acids off the cancer cell, it will be unable to shake hands with any Siglec receptor and therefore unable to deliver the inhibitory signal to immune cells through that pathway. So the actual molecule that we made to test this hypothesis, this was kind of a prototype drug, was a protein that was part monoclonal antibody. That's the parts that are in pink here. This is a ribbon diagram of a protein. And also part enzyme, where the enzyme is an enzyme called a sialidase. And there are known sialidase enzymes in the world. This is a family of enzymes that literally cut like a pair of scissors, like a blade. They will cut the bond between the sialic acid sugar, that pink diamond, and the tree that it's attached to. They'll literally cut that bond and the pink diamond floats away. So in this prototype, the antibody part was actually a known cancer drug called trastuzumab. It's the active ingredient in a breast cancer medicine called Herceptin. And that's an antibody that attaches itself to a protein that is abundant on certain cancer cells, and it's a protein called HER2. So this was a well-known drug, and we knew that it would park on cancer cells. And what we did is we attached to it a sialidase from a bacterium that we work with, which would cut the sialic acids. So the pink part is the parking part, and the green part is the blade. By the way, as was introduced, I was recognized with the Nobel Prize in chemistry back in 2022. And that prize actually was not to do with the biology of sugars at all. It actually had to do with a chemistry that we invented, which has the technical term bioorthogonal chemistry. And I bring that up now because we used that chemistry to attach the two proteins to each other. So bioorthogonal chemistry is how we connected the pink antibody part with the green enzyme part. And here are some chemical details for anybody who's interested in that. But to make a long story short, we were able to make this antibody sialidase fusion protein. And then we tested this in animal models of breast cancer. And here's some data from one of those models. This is a model where we take mouse breast cancer cells that have the HER2 protein on them. We graft those cells into the mammary fat pad in a mouse. So we're basically growing breast cancer cells in the breast tissue of the mouse. And then at different time points, we inject into those mice different reagents. And two of the mouse groups were injected with the antibody sialidase fusion, that's trastuzumab sialidase or TSIA, whereas other groups of mice were injected just with a salt water solution. PBS is what we call it. It's just like called the vehicle, it's like the solution the drugs would be dissolved in. And some of the mice were injected with the naked antibody on its own, so just the pink part of the antibody without the green sialidases. And what you can see here is that whereas these mice had tumors growing and getting larger and larger and larger as the days and weeks went by, and those mice had large tumors if they were injected with just the saline solution or the naked antibody. But the tumors were much smaller if the animals were injected with the antibody sialidase conjugates. And when we surgically removed these small tumors and looked at the cells in the tumors, what we found is the tumors were mostly made up of immune cells, and there were really no cancer cells left. So the immune cells had infiltrated the tumor, and those cells were killing the cancer cells. So it was really just almost like a scar and not a tumor. So this got us really excited, and ultimately we decided that we would try now to take this concept out of the laboratory and into a setting where we could make a medicine to test in human cancer patients. And so what we did, which is something that is, I think, pretty easy to do at Stanford, is we launched a startup company. And the company's name is Paleon Pharmaceuticals. And Paleon licensed the patents from Stanford. And these were patents that my students were co-inventors on, the students that did this work in my lab. And the scientists at Paleon kind of reconfigured the molecule to make it better suited for a human medicine. The important changes that they made is, whereas we had made a chemical conjugate, or we chemically connected the green enzyme with the pink antibody, the scientists at Paleon figured out how to make that connection between the enzyme and the antibody using all genetic approaches, so no more chemistry necessary. It just makes the molecules easier to manufacture at large scales for clinical trials. They also swapped out the bacterial sialidase that we had been using simply for pragmatic reasons with a sialidase that actually comes from humans. And it's always better if you're going to make a drug and put it into humans, if the parts are more human-like, because the body tends to accept the drug a little better. So this molecule that the Paleon team calls the eagle molecule, because the shape looks a little bit like an eagle if you squint and look at it, is actually going to be tested in humans starting in the second half of this year, 2025. It took about 10 years for us to get all the way from the science to the company to the drug candidate and to the clinical trial. But we'll find out within the next few years whether our hypothesis is correct and whether we can bring benefit to humans with these new next generation immune therapy candidates. So with that, let me just give you the take-home messages. First of all, I started out by telling you, in case you didn't know, all of your cells are coated with sugars. Second, it turns out the sugar patterns change on cancer cells. And one of those changes is that they have an abundance of that sugar called sialic acid. Now, based on the research from our lab, we know why that is. It's because when a cancer cell can overproduce sialic acids, it can engage and shake hands with the Siglec receptors on immune cells, which then inhibits the ability of the immune cell to recognize it as a cancer cell and to kill it, what we call immune surveillance. And then finally, by learning that, we were able to develop a new type of immune therapy that is basically a lawnmower that cuts the sialic acids off the cancer cell and makes those cells more susceptible to killing by the immune system. And we're going to test this in human cancer patients later this year. With that, thank you very much. I owe a huge debt of gratitude to the many coworkers who have been in my lab working on these projects. They include PhD students, postdoctoral fellows, staff members, and most importantly, a continuing pipeline of talented undergraduates, some of whom hopefully in the future will be represented by these families on this call. And with that, I'll open the floor to questions. Thank you.
Thank you so much, Carolyn. That was amazing. You made such a complex topic so easy that even I could understand it. So if you have any questions for Dr. Bertozzi, please put it on the chat line. Meanwhile, we already have a few, so let me pose them. So actually, I have one of my own. You talked about how drug development goes from basic research at universities like Stanford to drug companies. How does this really work together? How does this happen?
Yeah, it's a process that starts with making a discovery in the lab that I think, or my students think, and we all collectively agree, might have some commercial value. And if we think that that's true, what we'll do is we'll file an invention disclosure with the Office of Technology Licensing at Stanford. It's called the OTL. And what we're doing is telling Stanford, we think we discovered something. We think it could be valuable from a commercial standpoint, and we would like to protect that discovery with a patent. And in the United States, the rules are that universities own the patents, but the scientists who did the work, that would be me and the students in my lab, we are the inventors on that patent. But Stanford owns it, and Stanford can then license that patent to a company. And so what happens is we file the invention disclosure. Stanford evaluates it. If they agree that it could be valuable, they'll pay to have a patent filed. So now we've protected our intellectual property with a patent. Then I can start a company outside of Stanford that negotiates with Stanford for the license for that patent. And then if my company can get the license, if they can agree with Stanford on the financial terms, now my company has the exclusive license to that patent, and my company has the protection to try to make a drug. But I still have to raise money for the company so the company can hire people and continue the science and develop the drug. And that doesn't have anything to do with Stanford. That basically has to do with me trying to convince outside investors to put money into the company. But if there's ever value that comes from that patent, like if the company is successful, if Palion is able to make a medicine and the medicine eventually brings in revenue, some of that revenue comes back to Stanford based on the agreement on the license.
Yeah,
And
because
also
they
some of
own the
that
tech.
revenue comes back to the inventors, including the students who did the work. Yeah.
Great. Thank you. And one question was, does this research apply to animals such as pets like dogs and so on?
Well, yes, it does. In fact, what's interesting is there is some old literature from the 1970s where people had a suspicion that sialic acids might have something to do with the immune system. But nobody understood how exactly, because this is before we knew about the Siglec receptors. They hadn't even been discovered yet. But there were some inklings about this. And there was a study where people were trying to figure out if they could vaccinate against a cancer. And they were testing this in dogs. So there's a publication we found from the 1970s where a dog tumor was removed, treated with a sialidase, and then the tissue was put back into the dog to see if they could induce an immune reaction against the cancer tissue to help protect against future recurrences of that cancer. And it was a successful study, but they had no idea why, removing the sialic acids was making these tumor cells immunogenic. But that was a study done in dogs, and they cured some dogs of cancer. So I have a feeling that the same mechanism that allows cancer to progress in humans might also apply to animals. And, I had two cats die from cancer. Cancer is actually a very common thing in domesticated dogs and cats. And a lot of dogs, people's pets, lose their lives to cancer. And sometimes actually dogs are treated with the same cancer medicines that were approved for humans, and they just off-label use it for dogs too. So there might be a day when these immune therapies, if they succeed in humans, could be used in veterinary practice, not
Right.
inconceivable.
So important question. What does it mean for us as for our diet? I mean, I know you talked about sugars. Is it really that simple that we should avoid sugars and
That's very controversial. There's a lot of different facets of sugar biology. And so the sialic acid part of sugar biology probably just relates to the immune system. I have no reason to think that there's a link to our diet. But then there's other types of sugar biology that are very directly related to the diet, such as, the idea of low carb diets, right? A low carb diet or a low sugar diet is meant to affect your metabolism because there's a branch of sugar biology, which is really metabolic biology, and it has to do with the biology of a sugar called glucose. Glucose, everyone's heard glucose because people who are diabetic are testing their blood sugar for glucose, right? Glucose is the major food sugar that gives us energy. And there is a pretty compelling body of literature showing that cancers can often be fueled by sugar more so than normal tissues. Some people call this, it's called the Warburg effect, actually, named after Otto Warburg, who was a very famous German scientist from the 1930s and 40s who first discovered that tumors can be addicted to sugar. And so I think, there's some compelling literature suggesting that a person with a solid tumor, like a breast cancer or colon cancer, might limit the growth of that tumor by switching to a very low sugar diet, a very low carb diet. I've seen those kinds of studies. mostly in animals, and I don't know whether there have been really good human clinical trials around this, but in animal models, I think there could be a there there. Yeah. But it's kind of a separate biology from the sialic acid biology.
Right, right. Yeah, sugar is not good for many other things as well. So,
Yeah.
uh, so here's a question on a clinical trial that you did for, was it only for that type of cancer or was it for all types of cancer?
Our first clinical study will be focused on patients that have the right part for the lawnmower to park on, which is not every type of cancer, but just cancers that have the right, we call it a marker, the right marker for that particular antibody to park there. So for example, if you make a sialidase fusion with that antibody that binds HER2, then you could use that for patients that have cancers with the HER2 marker. So it's not going to be every cancer patient, and we might have to make different antibody sialidase fusions for different types of cancer,
Okay.
right? So it's kind of a modular platform where you can take the green enzyme and attach it to different pink antibodies for different types of cancer.
Okay. So I'm not sure I understand this question, but I'd pose it anyway. Is there any such thing as a preventive drug to keep cancers away?
Wow. Yeah, that would be the holy grail, right? Um, and right now, I think the most exciting, though very early stage idea is in the area of vaccines, right? Could we vaccinate against cancer the way that we vaccinate against measles or should be vaccinating against measles? Um, and there's some pretty compelling approaches to this, especially now because of all the advances in genomics and precision medicine. We've been able to identify specific genetic signatures for different kinds of cancer. For example, we now know that pancreatic cancers often have a particular genetic mutation. Like more than 50% of human pancreatic cancers have this one mutation. And if you could vaccinate people against that mutation, in principle, you could prevent pancreatic cancer from developing because as soon as a patient happened to get that mutation, boom, the immune system is ready with a sledgehammer to just beat it down, you know? So that's where I'm kind of watching. That would be a preventative strategy. But until we figure out all the science, and there's a lot more research to do for that, until we figure all that out, I think your best bet is the preventions that we know about. Don't smoke, use sunscreen. Um, we can vaccinate against HPV, which causes cervical cancers. And in fact, there's been a huge dip in cervical cancer frequencies ever since Gardasil was approved. So that is a cancer that you can vaccinate against because it's caused by a virus. So those things we can already do. But for other cancers, we're still very, very early stage.
Okay. So what differentiates the patients that respond to these treatments versus those who don't?
So we're trying to figure that out. That is like a $1 billion question right now. And I can tell you from some early clinical data, the team at Palion has been able to get biopsy samples from patients who were treated with those first generation checkpoint inhibitors. So remember, I showed that graphic from the metastatic melanoma trial where 50% of the patients survive, but the other half do not. So we got tissue samples from survivors and not survivors, and we analyzed them for sialic acid density. And what we found is that the people who survive tend to have lower sialic acids, and so probably less Siglec handshaking. And the patients that don't survive have more sialic acid and more
Mm.
Siglec handshaking. So our hypothesis is that the people who didn't respond to the first generation therapeutics are the people where the Siglecs are playing a more important role in the immune
Okay.
suppression. And so those are the patients that you might treat with a sialidase drug, a lawnmower drug. And maybe what you want to do is combine both the first generation therapies with the lawnmowers. Then you get
Mm.
more patients across the board. And in fact, in our clinical trial that starts this year, we do have an arm of that trial where the patients are getting a combination of a PD-1 blocking antibody together with the lawnmower therapeutic.
Yeah. And a question from a parent, how does a Stanford undergrad get a job in your lab?
Well, the first thing they should do is take some classes in chemistry and biology, just so that they have the basics. And then usually they just email me, and hopefully I have an open slot where I can accommodate them. Space is always the issue for every lab, and but I've always tried to reserve between 6 or 8 lab spaces for undergraduates, and they kind of rotate through. Usually when they come to me, they're, end of their sophomore year, beginning of their junior year. So they've had enough coursework where they kind of know that they're interested in the science. There's so many amazing labs you can work in at Stanford. And the student, has to figure out, what am I interested in? And if they get interested in this kind of science, then they'll come to me, we'll chat, talk about it. I'll have them meet with some PhD students and postdocs in the lab, because those are the people that would be with them at the bench every day. And see if they feel like, it's a good fit for them. And then I do my
Right.
best to make the space for everybody.
And here's a comment. You've been a great mentor, and wondering if you can talk about your experience as a mentor and role model for students.
Oh, well, I try. Yeah. Um, when I think about, how I can be the most effective mentor for students that work with me, usually I have to kind of try to turn my brain back to when I was in college. And what did that feel like? And just remembering what it feels like to be in a setting where you're in the early stages of learning and mastering the material. And most of what people are talking about is flying over your head. You are trying to manage a lot of, you know, demands on your time when you're a student. You have lots of classes you're taking, and there's lots of homework and tests and stress points and extracurricular interests. And then you're working in a lab, maybe in my lab, and just try to remember, what does that feel like? And, what are the ways that I can make students feel supported, as they're dealing with all the things that fly at them, especially when they're undergraduates. And that means, trying to give them a challenging problem to work on, try to give them enough independence so they can make their own decisions and feel like they're in control of their own research. Try to give them enough course corrections when I see them maybe heading off track, so that they don't get really frustrated with too much failure. Um, and inevitably, there's a lot of failure in science. It comes with the territory, and making it known to them that that's totally normal, and that there's, in fact, I would say some of the undergraduates who've worked in my lab, doing research in my lab is the first experience they've had with failure. These are very
Right.
successful
These are
kids.
smart students.
Yeah, they're so successful. They had 4.0s in high school, and they were captain of their football team or their debate club or their math team or principal violinist. They're so talented and so successful. They got into Stanford, and here they are. And even at Stanford, they're taking classes and probably doing well, studying hard. And then they get to the lab. And all of a sudden, instead of mastering knowledge that is already known to the world, they are now trying to create new knowledge that isn't known to the world. And they cannot anticipate what the outcome will be of their experiments. That is the point, right? So most of the experiments they do, they can't figure out what's going on. It makes no sense. It doesn't fit their hypothesis. It doesn't work. It doesn't even reproduce when they do it twice in a row, you know?
Yeah.
This is reality, and that can be a harsh learning experience, but it's more like the real world. So
Yeah.
I like to think I'm preparing them for the inevitable experience of failure in the real world, which
Yeah.
everybody will have to endure at some point.
Well, that's a great closing for this talk. Thank you so much, Carolyn. It was an amazing presentation, and you made such a difficult topic seem so easy that we could all get something out of it. And hopefully, this will also help us in how we lead our lives. Uh, I have some good news for you. We will get you 2 jars of our famous Stanford apricot jam.
Oh,
Uh,
yay.